This video is about pH, dissociation of water, dissociation of weak acid, Henderson-Hasselbalch equation, and electrometric determination of pH, pH meter, pH value calculation, and importance of buffers in biology. We will be learning all these topics and we will be just having a quick recap about all this. So let's start. So first, water. Water is a polar molecule consisting of 2 hydrogen atoms and 1 oxygen atom. We know that because of the high electronegativity of the oxygen atom, the electrons in the covalent bonds are unequally shared, creating a partial negative charge on the oxygen atom and partial positive charge on the hydrogen atom. This makes water a good solvent for polar and ionic compounds. Water molecules can also undergo a process called dissociation, in which they split into hydrogen atoms H plus and hydroxide ions OH minus. This process is represented by following chemical equation, where H2O gives H plus plus OH minus. The dissociation of water is an equilibrium reaction, meaning that both the reactants, water molecules, and products, hydrogen and hydroxide ion, are present in a state of dynamic balance. The concentration of hydrogen ions and hydroxide ions in pure water at a room temperature is equal, with the concentration of approximately 1 × 10⁻⁷ moles per liter for each ion. The dissociation of water is an important process in many biological and chemical reactions. For example, the pH of a solution is determined by the concentration of hydrogen ions present, and the acidity or basicity of a solution can affect the rate and direction of chemical reactions. In addition, the dissociation of water plays a role in the maintenance of acid-base balance in the body, as well as in the regulation of pH in cells and tissues. The dissociation of water can be affected by various factors such as temperature, pressure, and the absence of other ions and molecules. For example, increasing the temperature of water can increase the rate of dissociation, while adding acids or bases can alter the equilibrium position of the reaction. In summary, the dissociation of water is a process in which water molecules split into hydrogen ions and hydroxide ions. This process is important in many biological and chemical reactions and is influenced by various factors. Next, dissociation of weak acid. A weak acid is an acid that only partially dissociates in solution. In contrast to strong acid, which dissociate completely into ions in aqueous solution, weak acids only partially dissociate, resulting in a mixture of undissociated molecules and ions. The dissociation of a weak acid, represented by the general equation HA gives H plus plus A minus, is an equilibrium reaction in which the acid molecule HA donates a proton H plus to water and form hydronium ion H3O plus and the conjugate base A negative A minus. The extent of dissociation of a weak acid is determined by its dissociation constant Ka, which is a measure of the acid's tendency to dissociate in solution. For example, consider the dissociation of acetic acid and a common weak acid found in vinegar, and the CH3COH plus H2O gives CH3COO minus plus H3O plus. CH3COOH plus H2O gives CH3COO minus plus H3O plus. The dissociation constant for acetic acid Ka is approximately 1.8 × 10⁻⁵. This means that at equilibrium, the concentration of undissociated acetic acid is much greater than the concentration of the H plus and CH3COO minus. This means that at equilibrium, the concentration of undissociated acetic acid is much greater than the concentration of H plus and CH3COO minus. The equilibrium can be described by the equation Ka is equal to H plus into CH3COO minus divided by CH3COOH concentration, where H plus and CH3COOH minus and CH3COOH concentration represent the concentrations of hydronium ion, acetate ion, and undissolved acetic acid respectively. The dissociation of weak acids has important implications in many areas of chemistry and biology. For example, weak acids are often used as buffers to help maintain a pH of a solution. A buffer is a solution that resists changes in pH when small amount of acid or base is added to it, so weak acids can be used as buffers. The buffering capacity of a weak acid depends on its concentration, the dissociation constant Ka, and the concentration of the conjugate base N negative. In summary, the dissociation of a weak acid is a reversible reaction in which the acid molecule partially dissociates to form hydronium ion and the conjugate base. The extent of dissociation is determined by the dissociation constant Ka of the acid and has important applications in many areas of chemistry and biology, such as the use of weak acids as Buffers. Next, Henderson-Hasselbalch equation and its importance and applications. The Henderson-Hasselbalch equation is a mathematical expression that relates the pH of a solution to the pKa of an acid and the ratio of its conjugate base to acid forms. It's an equation of a mathematical expression that relates the pH of a solution to the pKa of an acid and the ratio of its conjugate base to acid forms. It's about half a page, so just pH is equal to pKa plus log A negative concentration by HA concentration. pKa plus log of A minus by HA concentration. pKa plus log of A minus by H concentration of HA, where pH is the measure of the acidity or basicity of the solution, pKa is the dissociation constant of an acid, and HA negative by HA is the ratio of the concentration of the conjugate base to the acid form. The Henderson-Hasselbalch equation has important applications in many areas of chemistry and biology, including acid-base equilibrium, buffer solutions, and physiological processes. One of the main applications of the Henderson-Hasselbalch equation is the preparation and characterization of buffer solutions. A buffer is a solution that resists changes in pH when small amount of acid or base are added to it. The pH of a buffer solution is determined by the dissociation constant pKa of a weak acid and the ratio of its conjugate base to acid forms. By using the Henderson-Hasselbalch equation, the appropriate ratio of acid to conjugate base can be calculated to prepare a buffer of a desired pH. In biochemistry and physiology, the Henderson-Hasselbalch equation is used to describe the pH-dependent behavior of proteins and other biological molecules. Many biological processes depend on maintaining a specific pH range, and buffers are often used to regulate pH in biological systems. For example, the pH of blood is tightly regulated around 7.4, and the bicarbonate buffer system plays a key role in maintaining this pH by balancing the concentration of carbonic acid and bicarbonate ions. Overall, the Henderson-Hasselbalch equation is equal to pH is equal to pKa plus log of concentration of A minus by concentration of HA. It's an important tool for understanding acid-base equilibrium and the behavior of buffer solution. Its applications in many fields of chemistry and biology highlights its importance in understanding the fundamental process that govern the behavior of living systems. Next topic is the electrometric determination of pH. Electrometric determination of pH is a widely used method for measuring the acidity or basicity of a solution. The technique involves the use of a pH meter, which measures the potential difference between two electrodes immersed in a solution. The electrodes are usually made of glass and are sensitive to changes in hydrogen ion concentration. The pH meter is calibrated using standard buffer solutions of non-pH values, and the potential difference between the electrodes is recorded for each buffer solution. Once the pH meter has been calibrated, it can be used to measure the pH of an unknown solution by immersing the electrode into a solution and recording the potential difference. The pH meter converts the potential difference into a pH value using the Nernst equation, which relates the potential difference to the concentration of hydrogen ions in the solution. The pH meter provides a quick and accurate method for measuring pH, and it has a wide range of applications in chemistry, biology, and environmental science. For example, pH meters are used to monitor the pH of wastewater, drinking water, and soil samples to ensure that they meet regulatory standards. In addition, pH meters are used to measure the pH of biological fluids such as blood and urine to diagnose medical conditions. Overall, the electrometric determination of pH is an important technique for measuring the acidity or basicity of a solution, and its applications is critical for ensuring the quality and safety of many products and processes. pH meter, parts and applications and working. A pH meter is an electronic device used to measure the acidity or basicity of a solution. It consists of three main parts: a glass electrode, a reference electrode, and a meter. The glass electrode is a sensitive electrode that responds to changes in the hydrogen ion concentration of the solution. It's made up of a thin glass membrane that is sensitive to pH changes and a silver wire that acts as a reference electrode. The glass electrode generates a small voltage in response to changes in pH that is detected by the meter. The reference electrode is a stable electrode that maintains a constant potential and is used as a reference point for the glass electrode. It is typically made up of a silver chloride wire and a solution containing a known concentration of chloride ions. The meter is an electronic device that measures the potential difference between the glass electrodes and the reference electrode are converted into pH values. The meter also has a display that shows the pH values of the solution being measured. The pH meter is a wide range of applications in chemistry, biology, and environmental science. It is commonly used to measure the pH of solutions in industries such as agriculture, food processing, and pharmaceuticals. In environmental science, pH meters are used to measure the pH of soil and water samples to access their quality and suitability for various applications. In addition, pH meters are used in research laboratories and medical settings to monitor the pH of biological fluids such as blood and urine. The working principle of a pH meter is based on the fact that a glass electrode generates a small voltage in response to changes in pH. The voltage generated by the glass electrode is proportional to the pH of a solution being measured. The reference electrode, which is immersed in a solution containing a known concentration of chloride ion, maintains a constant potential and serves as a reference point for the glass electrode. The meter measures the potential difference between the glass electrode and the reference electrode and converts it into a pH value using the Nernst equation. In summary, the pH meter consists of a glass electrode, a reference electrode, and a meter. It is an important tool for measuring the acidity or basicity of a solution and has a wide range of applications in various fields. Its working principle is based on the fact that a glass electrode generates a small voltage in response to change in pH, and the meter measures the potential difference between the glass electrode and the reference electrode and converts it into a pH value. Next, let's learn the structure of this pH meter. So this is the structure of parts of a pH meter. We can see that saturated calomel electrode is there, a filling port is there, solution of unknown pH, and then the ends are connected to the pH meter. Glass electrode is there, wax or gel insulation is there, heavy walled glass and AG wire, 0.1 molar HCl saturated with AgCl, magnetic stirrer. Then there is another pH meter electrode, KCl solution, filling port, AgCl reference electrode, and a glass frit. So this is the structure of a pH meter. So when we just try to learn the pH value calculation, the pH value is a measure of the acidity or basicity of a solution and is defined as negative logarithm base 10 of a hydrogen ion concentration in moles per liter in the solution. The formula for calculating the pH value is pH is equal to negative log of H plus ion concentration, where H plus is the concentration of hydrogen ions in the solution. For example, if the hydrogen ion concentration of a solution is 1.0 into 10 raised to minus 7 molar, then the pH value can be calculated as follows, like pH is equal to negative log 1.0 into 10 raised to minus 5 is equal to 5. Therefore, the pH value of the solution is 5. For example, if the hydrogen ion concentration, we just got 5, and therefore the pH value of the solution is 5, which indicates that it is slightly acidic. It is important to note that the pH scale is logarithmic, which means the change of 1 unit in pH represents a 10-fold change in hydrogen ion concentration. For example, a solution of a pH of 4 has 10 times the hydrogen ion concentration of a solution with a pH of 5 and 100 times this concentration of a hydrogen ion of a solution with a pH of 6. Additionally, it is important to consider the temperature of the solution when calculating the pH value, as the dissociation constant of water, which is used in the calculation of pH, is temperature dependent. So whenever we just calculate the pH, just remember that the dissociation constant of water is depend on the temperature. So calculate accordingly. Then buffer. Importance of buffers in biology in detail. Buffers are a class of chemical compounds that play a vital role in maintaining a pH of biological systems. pH is a measure of the concentration of hydrogen atoms in a solution and is an important parameter that affects the chemical and biological properties of the solution. Maintaining a constant pH is critical for many biological processes such as enzyme activity, DNA synthesis, and protein folding. Any fluctuation in the pH can lead to denaturation of proteins and other biomolecules, which can ultimately affect their function. Buffers are chemical compounds that can resist the changes in pH. When an acid or base is added to the solution, they work by absorbing or releasing hydrogen ions to maintain a constant pH. In biological systems, there are various types of buffers, including bicarbonate, phosphate, and proteins. Bicarbonate buffer is one of the most important buffers in biological system. It is involved in maintaining the pH of blood and other bodily fluids. Carbon dioxide is converted to bicarbonate in the blood, and the bicarbonate acts as a buffer to maintain the pH in the range of 7.35 to 7.45. Any significant deviation from this range can result in an acid-base imbalance, which can be life-threatening. Phosphate buffer is another important buffer in the biological system. It is present in many biological fluids such as cytoplasm, blood, plasma, and urine. Phosphate buffer system plays a vital role in the regulation of pH of these fluids, ensuring that the pH is maintained within a narrow range. It also helps to maintain the pH of intracellular compartments such as lysosomes and endosomes. Protein buffers are another essential class of buffers in biological system. Proteins are amphoteric molecules that contain both acidic and basic groups. These groups can absorb or release hydrogen ions depending on the pH of the solution. This property makes proteins excellent buffers that can maintain a pH of a biological fluid. The importance of buffers in biology cannot be overstated. They play a crucial role in maintaining the pH of biological fluids, which is necessary for the proper functioning of many biological processes. Any imbalance in the pH can lead to significant problems such as acidosis or alkalosis, which can be fatal. In addition, buffers play a vital role in many laboratory experiments such as DNA sequencing, protein assays, and cell culture. In these experiments, the pH must be precisely controlled to ensure the accurate results. In summary, buffers are essential compounds that play a vital role in maintaining the pH of biological fluids. They work by absorbing or releasing hydrogen ions to resist changes in pH when an acid or base is added to the solution. In biological systems, bicarbonate, phosphate, or protein buffers are the most important buffers. Their importance in maintaining the pH of biological fluids and laboratory experiments cannot be overstated. So, which are the major buffers? Bicarbonate, phosphate, and protein buffers.